My name is Tyler Stack. I am a PhD candidate officially I'm in the molecular biosciences department, and I'm going for a PhD in biochemistry. I've been at UT now for just over 5 years. And I work in Dr. Chris Whitman's lab, and he's a professor in medicinal chemistry and a pharmaceutical.
What kind of research do you do?
So, in the grand scheme, what my lab does is we study a family of proteins that are called the 4-oxaloprotonate tautomerase family. So it's just a group of proteins that are similar in structure, but they do different things in vivo. So our goal now is that we've entered sort of this genomic age. We have tons of DNA sequences, but we don't. So now we can find all these proteins that we didn't know about before. We don't know what they're doing. So the goal now is to see what proteins exist in nature and find out what they're doing. And then that should eventually lead into how can we design proteins to do something novel that currently nature doesn't do. So I specifically am looking at a few of these enzymes, which we sort of just call 4OTs. And these are involved in biosynthetic processes that are made for building up something large. So I'm looking at 4OTs that are involved in making some antiviral and antibacterial and some anti-tumor candidates that hopefully enter drugs. There's some analogs that are drug trials, and there's some that are currently being used as drugs in the clinics and things like that. And the goal is to, the end goal, nothing that I will ever do, is to, if we know what the proteins are doing and what sort of chemistries they can do, then we can either change them or feed them something unique, so that way we can make a different antibiotic or a different anti-tumor compound. And that makes chemical synthesis easier, because you just grow up these bacteria, feed them certain weird, exotic starting materials, and now it pops out some very insane and complicated structure that would be difficult to do by chemical synthesis. So that's kind of like the end goal. But my goal for right now is to figure out what these proteins are doing, so someone else can do the heavy lifting later.
Cool. What sparked your interest in biochemistry?
So when I was an undergraduate, I was mostly interested in organic chemistry. So I was preparing to go to graduate school, and I was going to do polymer chemistry, and I was going to make unique monomers, and from those I was going to make unique polymers. And that was like my thought process. That's like when I was a junior or so. So I started taking all of the organic level classes that the grad students would be taking, and I was preparing to take the GRE and the subject test in chemistry, and part of it on the subject test is biochemistry. And I didn't have to take biochemistry in terms of my degree, but I was like, let me just take undergraduate biochemistry just so I can get a better subject score on the chemistry GRE. And my one little quick semester, I really did enjoy the course. And I was trying to do a green reaction. That was what my senior thesis was about. And there was already some enzymes that existed that could do this reaction, and it's in water, and it was in ambient conditions, and it was just using oxygen. And that was what I was trying to do as an organic chemist, trying to find a mild condition to do a reaction, but there's already enzymes that were doing that already. So that sort of sparked my interest in moving over. So I ended up applying to a few places, and some as being an organic chemist, hoping to go into polymer chemistry, and then a few as a biochemist, hoping to do more biosynthetic stuff. And I ended up coming from Minnesota down here to Texas to do biosynthetic stuff.
Cool. I guess the next question would be, how does biology tie into the research you do?
So everything that I'm dealing with is the molecular basis of the larger biology picture that we're looking at. So what I was sort of hoping to do when I came in here is usually, in a lot of the classic biology textbooks, you'll see that you'll have an enzyme or something, and the enzyme is a box with a name on it. And then there's like an arrow that goes through it, and this is what it does. So the goal is to sort of just sort of remove the mystery of the box and find out what's actually working inside. Because, when I was an undergraduate, my goal was to do a pretty simple reaction, but I was trying to do it instead of with something that was petroleum-based, to find a way to do it that was renewable. That was the whole point of my project. And finding these proteins, these enzymes that could do it, was something kind of amazing. So the goal is to figure out how they do it. And if you think of it in terms of chemistry, you think of the atoms and the makeup, and then you can go broad picture and think of then the enzyme, and then go broad picture and think of how to do all this inside of a cell, and then inside of a huge batch reactor or something like that. And then you more approach like the classic biology that you think of when you're dealing with a whole ton of bacteria or whatever. Me viewing biology is sort of the way that I do my chemistry. That's sort of how I use biology.
And so, how is your undergraduate education helpful? How is your undergraduate education helpful in what you do now?
It's an interesting interplay with some of my other colleagues and stuff, because I was prepared to do straight organic chemistry, and that's what I spent all my time doing as an undergraduate, was doing organic chemistry. And then I have a minor in math as well. So that was going to be more helpful when I was going to do polymers, because there's a lot of statistics and things that go on with how to make polymers. So I was focused on organic chemistry and math, but then coming here now, I didn't think there'd be an outlet for math of any sort, really, because now it's growing up cells and then purifying protein. But it's nice going from a biosynthetic standpoint, because now I can think of how does an enzyme do this, and I can think of my organic chemistry routes and think of reaction mechanisms and how things work. And then I've started to slightly dabble in X-ray crystallography, which is very, very math heavy. And it's the only application that I could possibly use where you have to think of imaginary numbers. We talk about going from real space to imaginary space. So it's kind of fun trying to find an actual application for some of those really, really weird things they teach you in math, and they say that there is a reason why we teach you this, and you're like, there's no reason I should ever need to learn about imaginary numbers. But there's actually one application I have to use that for. So that was very nice. I did come into school here with a bunch of biologists, and I had to take biochemistry and molecular biology, and that was a quick catch-up there where I had to learn. I didn't know what a ribosome was, so I had no idea what any of these things were. And everyone was complaining about, we have to learn about T7 phage again. And I'm like, yeah, that sucks. I hate that T7 phage. No idea what they're talking about as I got prepared. So it is nice, because I get to talk to and work with some synthetic biologists and then microbiologists. And then the real hardcore X-ray crystallographers, and we can help each other do things that we couldn't do on our own.
What exactly is X-ray crystallography?
So, if you remember classic Watson-Crick, Rosalind Franklin, that's sort of the most famous example of crystallography, where you have some crystalline material, they were using DNA, and you shoot X-rays, and then they got that weird X pattern diffraction. And their goal was to figure out, okay, so if you have something diffract, where the idea is that you have light hitting an atom and then it bounces off in some direction, whatever the shape of whatever you're hitting, that'll give you a characteristic look. So if you just have a single atom, you'll end up getting a circle. If you have atoms in a square, you get a grid, like a grid of atoms. And then the goal then was when they saw this diffraction and they got that kind of X pattern. The goal was to figure out, without knowing what DNA looked like, what in the world could possibly give you a diffraction pattern in an X pattern. That's the goal, is you shoot a crystalline material with X-rays, and then the X-rays will bounce off. And then the goal then is to figure out what the shape of your crystal could possibly look like to give you that diffraction pattern. And that requires heavy math. But thankfully it's easy that a computer can do it, or that you could do it, but you just have to do it 20,000 times, which is something that no one should ever have to do. So all the early ones, like the Watson Crick, Rosalind Franklin, that one was just sort of intuited, and you got the basic idea of the double helix from that.
That's cool.
Is there anything that you regret doing or not doing during your undergraduate life?
Anything that I regret or
anything you could change?
So, the one thing, it's not a terrible thing. I think it's a natural learning process. I remember talking about how my first semester as an undergraduate, I went to the University of Minnesota to be a chemical engineer, and I was positive that I was going to be a chemical engineer. And I remember saying like I know everyone changes their major, and that's not going to be me. Like, I'm so lucky that I'm coming in, decided on a major, and I know what I'm doing, and it's going to be great. And I got accepted into the honors program in the natural sciences program, so I was taking honors math, honors physics, honors chemistry. And I was just with these other people, which I really enjoyed, but it was difficult to focus and then learn how to study, and then doing like the bare foundations that you needed in order to progress wasn't the most exciting thing. So I don't know if it's a bad thing, but I definitely wasn't as focused or really enjoyed what was going on as you are like when you're doing like your few first semesters calculus. It's nothing that you don't really derive satisfaction from doing calculus. But then once you finally move up, and like now I'm at a part where doing like these calculations for X-ray diffraction, it's not exciting to do the math, but it's exciting because you know what the purpose is, and then seeing the final product is kind of enjoyable. So the one thing that was mildly upsetting is that I just had like minimal focus and drive when I was a freshman, and the goal was to realize that it is critical. And sometimes now I also TA, so when I was giving one of these lectures, I was trying to reach back. I'm like, so you guys remember from like introductory physics, remember this thing that you learned? This is where we can apply it now. So it's terrible, and I dreaded physics, but now like coming back and talking about all those classic things that you learned and how to reapply them, it was difficult as a freshman to appreciate that at all.
Do you have any advice for current freshmen?
Given that I was not excited about classes, it seems easy for me to say that you should try hard and have the motivation. But it was so difficult. But the goal is to spend the time dabbling, I guess is the best that I could say. Where there's a lot of options currently available to you, and you have so much time left that this is also the time to make mistakes and try other fields, because you don't know what could excite you. So learning any level of computer programming could help you almost in any field that you go to. Doing engineering, any sort of engineering, helps in terms of critical thinking. Even if you don't want to become an engineer, things like that are always helpful. And then it's nice being here working with biologists, but having a different background. So I do think it helps to have some diversification in your knowledge base. This is the time where I would try different classes. I had some idea, because I never would have done biochemistry if it wasn't that I thought that I should do it to improve my GRE subject score. That's the best that I could say, is that even though right now it does kind of suck, because it's all the core foundational things, the goal is to think of how to apply it, and hopefully all the different classes you could possibly take, just to see if you could possibly enjoy electrical engineering. You have no idea if you'd like that. Even though right now it sounds terrible.
Yeah, I have like a lot of friends who complain about double E.
Yeah, oh no, everyone complains about it.
Yeah.
I started off chemical engineering, and a lot of my friends were in chemical engineering, and eventually I broke off into just chemistry. my grandfather was an engineer. So I remember telling my grandma that I'm dropping the engineering, I'm just going to do chemistry. Her reaction was, are there jobs in chemistry? I'm like, I'll be fine. Don't worry about it.
Yeah.
I could have just said that I was doing some like Slavic studies or something, and she would have the same reaction. Yeah, I mean, it always gets tough and kind of scary once you get to the end. We have a lot of our undergraduates are in chemistry, and they'll always complain about, or biochemistry, and they'll complain about all of their upper-level courses. I complained about mine when I was an undergraduate. My chemical engineering undergraduate friends all complained about their classes. It always gets sort of scary but fun and intense once you finally get close to the end.
Alright, so what could undergraduates do to get involved in research?
There are some outlets for freshmen specifically to get in to the FRI programs. And then even after that, it's nice to get a research assistant to come in early, because if you have it, even like as a graduate student, when I came in and I'm looking for a lab to join for my entire PhD career, I don't know anything about what these individual labs are doing. There's still like an extreme learning curve to become proficient in the language and the techniques necessary to be good in any lab. So there's always going to be that scary part where you're like, I have no idea what you're talking about or what you're doing, because it's going to be true for anyone, basically, going to any new lab. So as soon as you feel like you'd like to and you have the time, it's probably a great idea to try to get in as early as possible, because if you devote time to a lab, the longer you spend, the better you'll get. And then professors generally don't like to train someone if they're only going to be around for a semester, so they expect a year or something like that. So I was told, like, once I start taking organic chem lab and I wanted to work in organic chemistry group under a professor, that's when I should tell them that, like, I'm learning the organic chemistry techniques right now. I'd like to join a lab. So I joined an organic chemistry lab shortly after I finished the course for organic chemistry. And I was there for 2 years, and I think I sort of appreciate that more. But even still, even if you show that you have some promise and you're excited about something, even if you have no idea exactly what they're doing, there's some aspect you can possibly learn something and get started. But yeah, the longer you're there, the more productive you'll be, and you might get something great out of it.
Was the organic chem then like first research you ever done?
Yeah. So I did zero research, and I must have been a sophomore, and I emailed probably 10 professors that were just listed like under the organic chemistry heading, and I asked if I could meet with them and talk about what the research was and if there was a position available for a graduate. I think 3 ignored me, and then I had 6 or 7 meetings with different professors where they were just there to meet and talk with me and just to see if I was interested in what they were doing. And the point is, like, I have no idea what they're talking about, right? So the goal is just to appear excited and then try to ask a question about what's going on, even though there's no way you can ask an intelligent question.
Yeah.
The advice that I always give is when you enter graduate school, at least for biology, there's usually a round of interviews when you're coming in as a graduate student. And the interviews are mostly formality. Because I apply to graduate school, you are invited for an interview. So they will pay for your airfare and then pay for your hotel and then wine and dine you for a couple nights. So they're paying a lot of money to show off the school. So if you're being invited for an interview, it's not really, are you going to make it or not? It's still mostly showing off, but then giving them the opportunity to say, you're insane in person. Like, you looked okay on paper, but you're crazy. So you can then formally get a refusal, right? So at the interview, you meet with some professor and they talk about their research. And students are always like, should I like read up on all of their articles? Should I find out what they're doing? Should I try and answer a smart question? And I'm like, that's a lot of work because sometimes you don't know anything about what they're doing. So the best thing is to have them talk about their research, and then you just ask, why do you think that is? That's kind of weird. Why would it do that? And then they love that question because they're glad to like expound upon their like hypotheses about why something is the way it is. So that's like the best advice I could give. Is you meet with them, and at least it should sound interesting. If at this point when they're talking to you, you sort of tune out, it's probably not a good sign. But if you'd like to join, listen, try your best to understand, and then the key question at some point is to go, that's really weird. Why do you think that is? Because that's like, that's the closest thing you can get to asking an insightful question without knowing a single thing about what they're talking about.
Just keep on questioning them, making interested.
Yeah. Because that's what they want to see. They want to see that you're interested and that you're motivated. Because right now they have no idea if you're skilled. Because you have no proof of that. But yeah, I think, I forget the exact numbers, but I think I emailed 10, I got 7 responses or so, and then I think 5 or 4 of them said that they'd be willing for me to join the lab or something. And I have no experience. The goal is just me talking with them and stuff. So that's the best you can hope for.
So you just email professors and
Yeah, and I gave them as much information as I possibly could. So I said, I am this year, here's the classes that I'm taking, here's my future plans. I think I gave like my GPA or my transcript even, just to show they could see everything. Just try to be super upfront. I don't know if you have to do that, but they may have appreciated that, I don't know. Because it's kind of all they have in terms of figuring out like where you stand. And as long as you mention some excitement and you seem excited when you meet them, that's the best thing you can do. Because they're not going to be interested in helping you if you don't seem interested or excited in what's going on.
Mm-hmm.
So, yes, like, since we should probably come to a close at the time, I guess we'll end it with what's your favorite Starbucks drink?
This currently, so today was officially the last class day. That next week after Thanksgiving is over, we're having one more class. We're just answering questions. So we went to celebrate the final thing. So this is a dirty chai, which is just a hot chai tea latte, but then they put espresso in it. And this is what this is, and it's great. I love this.
Nice. Well, thank you so much for doing this.
Thank you.
I really appreciate it. Great job.